Hello, everyone. Thank you for tuning in. We will be starting in a couple of minutes with our Mind at Home episode. So let's just wait for a few more of our friends to come join us and watch, to tune in before we start. All right, hello, everyone. So while we are waiting, I hope you guys have read the description of today's Mind at Home episode so that you'll have a general idea of what we are going to do this morning. Again, we'll be starting in a couple of minutes. You may call your family, call your friends to join us for today's Mind at Home episode. And thank you for spending your Wednesday morning with us here at the Mind Museum. And hello, everyone. We will start in a couple of minutes. Let's just wait for more of our friends to tune in before we begin. Hello. All right, we're starting in a little while. Tell your friends, share this video to your friends and your family so that we can have as much more people watching. Hello, Asami. How are you this Wednesday morning? All right. So again, hello everyone. Pleasant morning to everyone. My name is Mind Mover Mario. I'm one of the science communicators of the Mind Museum. And welcome to another episode of Mind at Home. I know that we are going through really trying times right now. The COVID 19 pandemic is making a lot of people sick, and it has put all of our lives on hold. And what are the things that we can do to cope during these difficult times? And what can we at the Mind Museum do to help you guys out? According to the US Centers for Disease Control and Prevention, taking the time to unwind and to do some other activities that you enjoy doing can help cope with the stress and anxiety brought by the pandemic. And we at the Mind Museum firmly believe that fun and learning can coexist, can go together, and that they can play a big role in coping with the stress of the present situation. In order to help in slowing down the spread of the disease and to flatten the curve, as they say, we have to stay at home as much as possible. And I know that in many places of the country, the stay at home orders have already eased up, they've already relaxed a bit. But remember that the virus is still out there. It can still spread, you can still get it, and you can still fall sick. So I hope you limit going outside only when it is really essential, and that you remember to wash your hands regularly with soap and water, especially when you've been outside and you've touched things that other people may have also touched. So remember to wash your hands regularly. It's very, very important. Now, how can we have a little bit of fun at home, right? Well, we are all stuck at home. How can we have a little fun? And is it possible to do this while we are learning? And of course, we can do that. We at the Mind Museum firmly believe that we can make fun and learning go together. With Mind at Home, we are going to show you some science activities and science experiments that you can do at home with the materials that many of us, already have. All right, so for this morning's Mind at Home, we are going to extract DNA from a living thing. Very, very exciting. We're going to do an experiment where we open up cells and try to extract DNA. It's one of the most important part of the cell, the DNA, and we're going to use techniques in chemistry. We're going to use chemistry in order for us to extract the DNA. We're going to apply uh, chemistry or techniques in chemistry with our knowledge of biology. Now, I remember the first time that I did this experiment was in the lab, not at home. I did this in the lab way back in my college years. I studied public health, and back then we extracted DNA from an E. coli. E. coli is a bacteria that you can find inside our guts, it's in our tummies, and you can find this as well in dirty water. So you shouldn't be drinking dirty water because E. coli can make you sick, or some strains of E. coli can make you sick. So what we did was we extracted DNA in a lab. It wasn't much, hindi siya madami, very konti lang yung na-extract namin na DNA because bacteria is a single-celled living thing. It's just one cell big. So maliit lang siya, right? So maliit na maliit lang yung bacteria. So now, before we start, let's have a little review on what cells are and what DNA is, and where can we find cells and where can we find the DNA. The cells is what we call the basic unit of life. It is the smallest level of a living thing. That means that all living things in the world, may it be a bacteria, may it be a plant, an animal, or even us humans, we are all made up of cells. And actually, the smallest living things that we know are just one cell big. So kasing laki lang sila ng isang cell. And that's why the smallest living things we know, we actually can't see them with our naked eyes. We have to look at them through the microscope because they're very, very, very tiny. And of course, more complex living things like us humans, plants and animals, are made up of much, much more cells. Actually, us humans, in fact, are made up of trillions of cells. So that's trillions with the letter T, right? So not millions, not billions, but trillions of cells. And aside from making up all living things in the world, cells are also containers para rin silang lalagyan. There are many things that are inside cells. Maraming bagay sa loob ng cells that are very important for their function. Parang maraming function na ginagawa ang cell sa ating katawan, and all those stuff inside them are very important. And for this morning, we're actually going to focus on just one of those things inside the cell, and that is the DNA. So DNA are found inside the cells. So what is a DNA? DNA is what we call a biomolecule. It's a molecule that is very important for biological processes to happen. So for us to stay alive, for living things to stay alive, we need these processes, and DNA is one of these molecules that we need. It is important for our cells to repair and maintain our bodies. So that's why when we get a sugat, when we get a scratch or a scrape, our bodies can heal itself because of our DNA. It helps the cells to repair our bodies. So a DNA is a collection of molecules called nucleotides. It's a chain of nucleotides, a very long chain of nucleotides that cells use. So these are a set of instructions, para silang instruction manual kung paano gumawa ng mas marami pang cells. So cells use this to make more copies of themselves, and as well as to make some of the very important products that is needed for life to happen, such as proteins. So we use our cells' DNA to make proteins. So what are the materials that we are going to need for our experiment this morning? How can we extract DNA from a living thing just by using things you can find in your kitchen cabinet, maybe in your bathroom, some of the things we're going to use. So here are, let me show you some of the things that we are going to need. Ayan. So hope you can see. So first of all, we need a living thing, something where we can get our cells and DNA from, and that would be the banana. We are going to use a banana because bananas come from where? Bananas come from plants, and plants are living things. So bananas have cells actually. So we are going to use a banana to extract DNA from the cells of a banana. Next, we need, I'm going to use a resealable bag. A resealable bag where I'm going to put my banana in later because I need to mash my banana. So we're going to need resealable bag. If you don't have a resealable bag, you can also just use a bowl. I have a bowl here. You can just put your banana in there to mash your banana. So kailangan ba saging? Kailangan ba banana yung gamitin? Well, not really. I'm just using the banana because it's soft. It's easy to mash with my hands, right? So I'm going to put it in the resealable bag later and going to mash it. So madali lang durugin yung saging with our hands. That's why I'm using a banana. Next, we are going to need warm water. Or I forgot my warm water. I have an assistant here at my side. I'm going to ask for warm water. We're also going to need warm water. It doesn't have to be hot water, just warm. Next, we are going to need liquid soap. I have here, so paubos na yung soap ko. This is just regular hand soap. This is what we use to wash our hands. So paubos na because we are washing our hands regularly. So konti lang naman, we just need around 2 pumps of warm water. So just 2 pumps of our soap. Sorry, just need 2 pumps of soap. We are also going to need salt. So this is just plain old iodized salt. We need around half a teaspoon of salt. So later I'll be explaining what these materials or what these ingredients are for. We are also going to need a strainer. So I have this. This is parang net strainer that I'll be using. It doesn't have to be a strainer like this. You can use maybe a coffee filter. If you have coffee filter at home, you can use that. If you have old handkerchief, mga lumang tela sa bahay that can sift through a mashed banana, pwede din naman yon. So, pwede din namang tela lang if you don't have a strainer like this. Next is, we are going to need cold alcohol. So, this is alcohol that I placed in plastic cups. Ayan. So, this is alcohol that I placed in a plastic cup that I put in the ref. So, malamig siya. Kita nyo, may mga condensations siya. So, kakalabas lang from the ref. So, this is just plain rubbing alcohol. The same rubbing alcohol that you can buy at the store, at the grocery. Yun lang din siya. So, later I will explain what this is for. And what else? I have a measuring spoon to measure my 1/2 teaspoon salt later. So, it doesn't have to be exact measurements. Hindi naman kailangan very exact measurements. Pwede na kayo magtansya ng experiment. So, this is my warm water. It's warm. I'll put that here. Right, so let's start our experiment. The first thing we're going to need, also you're going to need some extra cups to put your mixtures later. So, these are just recycled plastic cups. Right? Recycled plastic cups lang siya, preferably clear so that you can see the reaction as they happen. And I have a stirrer, which is a barbecue stick. Panghalo lang. The spoon is not really required, but I might need it. That's why I have it here. So the first step is to mash the banana. So this is my banana. I'm going to peel it open. And medyo malaki yung banana, so I'm not going to use the entire thing. I just need around half of it. Just half of the banana, and I'll save this as my snack later. Right, so I'll put the banana inside the resealable bag. I'll seal it, and then all you need to do is mash it with your hands. So that's why I chose the banana, because it's very easy to mash with your hands. So if you're going to use other fruits or other vegetables and it's not so easy to mash, baka you might need to use if you have a blender at home, you might need a blender pa. So this one, I can just mash with my hands. So why do we need to mash the banana? Why is it important na durugin yung ating banana? Well, we have to separate the cells from each other. And we can separate the cells by mashing them like this, or mashing the banana like this. So durugin nyo lang. Yan. So may nafe-feel pa akong mga small lumps. Again, we're doing this, we're mashing the banana because we need to separate the cells. Okay. So after that, just set it aside first. And we're going to make the next, one of the very important part of our experiment. It's what we call the lysis solution. So it's spelled Lysis, lysis. And what that means is to destroy the cell membrane. The cell membrane is the outer layer that covers our cell. So we need to destroy it in order for us to get inside the cell and remove the DNA. So how do we make a lysis solution? That is the use of our soap, our salt, and the water. First, in our cup, I'm going to put 2 pumps of our liquid hand soap. So 1, 2, I'll add 1 more. So as I told you, paubos na yung aming liquid soap. So konti lang. Next is we need the half teaspoon of our salt. So later on, I'll tell you, oopsie, I'll tell you what these ingredients are for. So half a teaspoon of our salt. Add that to our soap. And next is our water, our warm water, so that we can dissolve everything together. So this is around half a cup of water. That's about enough. And just stir it until you no longer see any grains from our salt. So pag wala ng salt grains, just stir it, mix everything together. So that's why also important to use warm water so that our salt can be easily dissolved. Ayan. I try to add a bit more soap. Dahil konti lang yung nalagay ko kanina. So for this much banana, siguro yung soap would be around 2 teaspoons. I just estimated it to be around 2 pumps. So parang 1 pump for 1 teaspoon of soap. So just a rough estimate. So kapag okay na yung ating lysis solution, what we are going to do next is we are going to pour it into our mashed banana. Nagkalat na ako, dami ng salt. So I'm going to pour it inside our mashed banana, like so. And what we're going to do is we're going to seal it. So I prefer a resealable bag para hindi mag-spill, hindi matapon yung ating mixture. Oops. Now what we're going to do is we are going to mix our mashed banana and our lysis solution together. So what this does is it opens up our cells Again, because soap can do that. Soap can cause our cells to open up. It destroys the cell membrane. It's actually similar to why we are being asked to always wash our hands with soap and water, because the soap can destroy the envelope of viruses, the cell membranes of bacteria that can be in our hands. So we're just going to make sure everything is soaked in our lysis solution. So just be careful para hindi masyadong maging mabula or foamy yung mixture. So when everything is mixed together like that, what we have to do next is we need to strain the mixture. That is what the strainer is for, because we don't want the lumps of banana to go into our mixture. All we want is the liquid part. So we're going to strain it. So if you don't have a strainer, again, you can just use a coffee filter or an old piece of cloth. Oops. Ayan. So we are going to strain it like so. So this part, medyo you need patience. You have to wait for the liquid to drip. And that is really what happens in a lab. A lot of times, you have to wait for things to happen. All right, so maybe this is a good time to explain all the other ingredients that we use in our lysis solution. So first, we use soap. Again, the soap is for opening up the cell, similar as to what happens to the virus when we wash our hands, right? We destroy the outer covering of cells and of viruses and bacteria. So that is what the role of soap is. Next is our iodized salt. So what is the iodized salt? Or, well, it doesn't have to be iodized. You just need salt. The salt is, it neutralizes the charge of our DNA, because DNA is a charged molecule. So it neutralizes the charge of the DNA, and that makes the DNA less soluble in water. What that means is it makes it less dissolved in water, because DNA is soluble in water. Nadidissolve siya sa tubig. So nadidissolve siya sa tubig, and by adding salt, you decrease that solubility. Binababaan natin yung pagka-dissolve niya sa tubig. I'll add a bit more. And again, the warm water is for just so we can bring the soap and the salt together. Yeah, so it's really, you really need patience to wait, because it's dripping very, very slowly. Yeah, so you don't have to use all of it. Tansya lang din. So this is just around, I don't know, 1/4 of the cup. So in the lab, we don't use soap and salt like what I did, right? So we don't just use stuff that we find at home. So in the lab, there is a specific lysis solution that they use. So it's a reagent called the lysis solution or lysis buffer that they add to their living thing whose DNA they want to extract. So these are pre-made solutions na that you can buy in chemical stores. Well, you can just buy it easily, I think. You have to have a license, or we have a permit in order for you to do that. So that's about enough. So this is now our filtered mixture of our lysis solution and our banana. Oops, let me just fix some of my stuff. So this is it. Next thing we're gonna put is our cold alcohol. Now, what is the role of the alcohol here again, this alcohol, just regular rubbing alcohol, 70% rubbing alcohol that I put in a cup and place in the ref for about an hour para lumamig. Nakita nyo, nagko-condense siya, it's cold. And what the alcohol will do is it will precipitate our DNA. So can you say that? Precipitate. All right, so precipitate is a process where we take something out of a mixture or out of a solution. So right now, we can't see where the DNA is, right? It's just liquid. So once we put our alcohol, our cold alcohol, it will precipitate the DNA because DNA is insoluble in alcohol. It doesn't dissolve in alcohol, unlike in water. That's why we need to add alcohol so that we can make the DNA go out of our mixture. So makita natin later if we are successful. So for this time, we have to pour it carefully because we want our alcohol to stay on top of the solution para hindi siya mag-mix together so that we can see clearly the DNA precipitating. So I'm going to pour it carefully. Just have to be careful. All right. Now, all we have to do is wait for our DNA to precipitate. Actually, I can already see some of the DNA precipitating. Kung nakikita niyo po yung parang bubbles or the white stuff that is going up, that is the DNA that is precipitating out. So I have here, so that you can see more clearly, I have a blackboard where I can put it against. Kita ba? Can you see it? All that's parang white strands or white thread that is coming up into our alcohol on top. That is the banana's DNA precipitating. Ayan. Ayan. All that white thread, parang thread na puti na umaangat, that is the banana's DNA. So all the stuff that makes the banana a banana, so from the banana's shape to its color to its taste even, it's all stored in that white thread that you can see coming out of our mixture and into the alcohol. Amazing, right? How something very, very, very small like a DNA we can actually see forming. So maybe I can also show you from the top. I hope it's clear. Is it clear? Can you see? All right, so I'll hold it here so that you can watch the DNA. So all those white stuff, parang fibers, parang cotton, that is forming, that is the DNA, or that is the DNA of the banana. And there we go. So that is the banana's DNA. All the instructions on how to make a banana is in there. Right, one more time. Let me show you this view. Take out my blackboard again. And all those white stuff that is floating in the alcohol, that is the DNA. Right? How cool is that, right? How we can observe something really, really, really tiny like DNA. But of course, this is not just DNA from one cell. This is the entire DNA of many cells, right? Actually, half of a banana. So that is a combination of many, many cells. Now, what can we do with this DNA? Well, in the lab, we can study this. We can look at it under the microscope. We can sequence it. That's one of the most popular thing that scientists do with DNA is they sequence it. And what that means is we read it. We read the DNA from end to end so that we can see how different one DNA is from another. So maybe my DNA from a banana's DNA, from a bacteria's DNA, or another animal's DNA, we can compare them together. And again, in our, right, so that white visible layer that is in the middle of the alcohol and our solution of banana and lysis solution, that white layer, that's actually all precipitated DNA. There's quite a lot, right? It's quite a lot of DNA from the banana. Right. So again, in the lab, this is not how we do it exactly, right? Again, there are steps that we have to do. It's this one, our experiment at home. It's very well. It's at home, so we're using just kitchen ingredients. It's very crude, but in the lab, we do this very very clean, very very exact, very very like methodical, but it's essentially the same thing. We use a detergent to lyse the cell membrane to make the DNA leak from our cells, and then we use alcohol to precipitate the DNA from the mixture. Okay. So now let me try to strain our mixture so that maybe I can show you some more the DNA. Let me bring back my strainer again. Oops. Just wipe some of the banana bits away. I'm going to try to strain my DNA. Let's see if we can strain it. Oh, there it is. Just wait for it to drip again. Maybe I'll add some more. So if you have any questions, it's time to type in your questions into our comment section. Can we still extract DNA? There's a question from Monique. She's asking if we can still extract the DNA from cooked fruits and vegetables. Well, I don't think so. Our DNA would have probably been cooked as well. So I don't think it's viable for extraction anymore. Let's see if I can scoop out some of the DNA. Meron pang alcohol bits, or meron pang alcohol stuck to it. Maybe I can show you like this. Just need to, and, oops, baka matumulo. There it is. All those white slush are actually fibers of DNA. I don't know if you can see it. It's the DNA of the banana. Now, how do we know that this is actually the DNA? If we can't check it under the microscope, well, a lot of scientists have done this experiment, and a lot of scientists have described what DNA looks like in real life, like this. So we know that it looks like fibers, white cottony stuff like this. So we know that this is our DNA. All right. So there you have it. That's how you can extract DNA from a living thing from the comforts of your home just by using materials that you probably already have at home. So if you have any questions, go type them down at the comment section. Let me back read for a while kung merong mga questions at the top that I missed. Oh, Harvey is asking if I used ethyl or isopropyl. Actually, I don't know. I have no idea. So that means you can just use any alcohol. I actually don't know if that was ethyl or isopropyl alcohol that I used. So you can use both ethyl or isopropyl. Kai is saying that they are going to do this later with her kids. That's great. So let us know what fruit you used or what living thing you used to extract your DNA from. Liquid hand soap or dishwashing liquid, any soap will do. I use liquid hand soap, but you can also use dishwashing liquid. You can probably use body wash. You can probably use shampoo even. As long as there is soap in there, it will work. In extracting DNA in animals, does it require living cells? Well, yes, the cells needs to be alive. They need to be alive for our soap to work, because the soap will actually be the one to kill the DNA, to kill the cell, right? Because as we open the cells up, everything leaks out, and that essentially kills the cell. And that's how we get inside the cell to extract the DNA. Any more questions? Asami is asking, can we use any kind of alcohol? He or she is saying, I have only cooked sake. Well, sake may be a little low on the alcohol concentration because this is at least 70% that I use. So yung mga drinking alcohol may be a little low on the alcohol concentration. I don't know, but you can try. You can try and you can share your results, because that's what we want, right? In science, we want all scientists to share their results, their findings in their studies, so that we have a big body of knowledge. So I hope I didn't miss any questions. Let me just go through our comment sections one more time. All right, so that's about it. I don't think. Are you seeing any questions? Wala na. All right. So if you do try this experiment at home, I hope you share with us what fruit or what cells you used. So from what maybe animal or plant. Maybe if you have uncooked meat at home, maybe you can get a part of it and blend it. Maybe, I don't know. So it would, I don't think it would work if you just have a teeny tiny amount. So that's why I use at least half of a banana, so that we have a lot of cells to extract the DNA from. So they actually are also doing this with the testing that we are doing for the coronavirus. As we swab from inside our noses, we get the cells from the back of our throat. We scrape the cells with the swab, and we actually use a lysis solution as well to open those cells up and see if there are bits of the virus inside our cells. But of course, it will not work. Our particular setup here, it will not work with that amount, teeny tiny amount of cells. So kailangan mas madami yung cells na ginagawa natin. Because in those kinds of tests for the coronavirus, what they do actually is they use a machine to amplify or to make more copies of the cell or the DNA. Pinapadami nila. So that's why they are able to see. For this particular setup, you will be needing quite a lot of cells to extract the DNA. So I hope you guys enjoyed our Mind at Home episode for this morning. Again, just a reminder to everyone to please stay at home as much as possible, even though the stay-at-home rules have been eased up at your place, at your area. Remember that still has a community quarantine in the name, right? So even if it's GCQ or modified ECQ, that still has the CQ. We're still at the community quarantine phase. We still need to stay home as much as possible because the virus is still out there spreading and can still cause a lot of people to get sick. Always wash your hands regularly with soap and running water. We have seen what soap does. It lyses the cell membrane and the envelope of viruses to open them up and kill them. So soap is very effective, and you should do it for at least 20 seconds. And while we are all staying at home, or you are at home, please follow the Mind Museum. We are at the Mind Museum on Instagram and Facebook. And on our Facebook page, you can watch all the past Mind at Home episodes. So from today, starting today, May 20 until the end of May, we are doing a series of biology experiments and activities. So if you are interested in biology, you can watch all of our remaining episodes for the month of May every Mondays and Wednesdays at 11 a.m. at the My Museum Facebook page. So feel free to tag us with your videos and photos if you do our experiments and activities. I want to see what you do. I want to see your finished products. Please stay safe at home. Thank you so much, everyone, and happy lunch. It's almost lunchtime pala. Thank you for watching. I'll catch you guys next time. Bye-bye.